Hello students, have you heard the word BT cotton, BT brinjal, BT soybean? What does this BT stands for? Do you know students, BT stands for Bacillus thuringiensis. It is a bacteria name. This bacteria found in the soil. It is a soil bacteria. In genetic engineering, with the help of rDNA technology, we are modifying this bacterial gene and introducing into the crops to get a good harvest. You know students, what happened in the agriculture field? These insects, bollworms, armyworms, flies, these different type of insects are attacking on the plants and reducing the harvesting. To overcome this problem, what these genetic engineering scientists have done, they are isolating this gene from the Bacillus thuringiensis bacteria and introducing into the crops to make genetically modified organism, GMO. And how this BT cotton, BT brinjal is differ from the other normal brinjal, normal rice? You know, these crops contain bacterial gene in their body. What is exactly mechanism? Now we are going to understand one of the complicated topic from 12th biology, that is applications of biotechnology in agriculture field. How can we construct the genetically modified crops like BT crops, BT brinjal, BT cotton like? Okay, so there is a very interesting concept is there. Now we are going to understand. Student, this is also very important for your competitive purpose and also CBSE examination. Let me take the name of the bacteria. What is BT stands for? I told you BT stands for Bacillus thuringiensis. Yes students, this is the bacteria name, Bacillus thuringiensis bacteria name. Let me take the bacteria. This is bacteria. Now, this Bacillus thuringiensis bacteria have a specific gene in their genome. So, I am taking a specific gene. This specific gene we called XII gene. This specific gene we call a XII gene. XII gene. This XII gene undergo transcription and translation to give a protein, a toxin. This XII gene we can also write toxin gene. Toxin gene. And this gene undergo transcription and translation to give a protein. The protein is called a XII protein. XII protein. When you're writing XII in the capital letters, that means you're talking about the protein, XII protein. This XII protein is poison. This XII protein is toxic substance, toxic protein. So here, this protein is toxic protein. This protein can kill insects like bollworm, armyworm, beetles, caterpillars, different type of insects. That's why we can say this protein is insecticidal protein. That means it can kill the insects. So this is exactly mechanism. So sir, I have a question. Why don't this toxic protein can kill the bacteria? Actually student, this protein is secrete in the form of a protoxin. That means inactivated toxin. So I can write here, initially it will secrete. Initially this protein is secreted in the bacteria, that is in the form of protoxic protein. That means inactivated, or we can also say it is inactivated protein. Inactivated protein. Okay, to convert this inactivated protein to activated protein, alkaline pH is required. That we come to know little later. So that alkaline pH is not there in the bacteria, so this protein cannot kill bacteria. Okay, so what scientists have done, so they understood this total mechanism. Okay, there is a gene, the gene is called CRY. So CRY is secreting, a CRY is undergoing transcription and translation to give a protein. The protein is a toxic protein. So what scientists have done, scientists isolated this CRY. This is CRY from the bacteria. I hope you get my point. This is CRY isolation. They are isolating this CRY from bacteria, and this CRY they introduce in the plasmid, Ti plasmid. So I'm taking here, it is with the help of Ti plasmid. This is plasmid. So here I'm writing Ti plasmid. We know what is Ti, tumor inducing plasmid. From which bacteria? Agarobacterium tumefacium bacteria. Now what happened? This gene, that CRY protein gene, they introduce into Ti plasmid. So they construct what now? Tell me here. When they introduce this CRY protein gene in the Ti plasmid, now this is, we can write rDNA, recombinant DNA. Right? So this recombinant DNA they introduce in the plants. So this recombinant DNA they introduce in the plants. In where? Plants. Just assume this is cotton plant. Okay? Cotton plant. So this CRY gene, now bacterial gene in the plant. So that plant we can say genetically modified plant or organism, because plant having bacterial gene. So in the plant body, what happened? If I'm taking this one, look at here, this is plant. So here in plant, different part of the body of plant, the accumulation of this protein takes place. What does it mean? In a plant, look at here. In plant, this CRY gene undergo transcription and translation to give a CRY protein, and this CRY protein accumulated in the all plants part, leaf, root, bird, everywhere of the plant, this CRY protein is accumulated in the plant. So look at here, this is a ball. Okay, cotton ball, just assume. So what happened? There is a caterpillar. Look at here, there is a caterpillar. This caterpillar is now attacking on this crop. So they started to feed on the balls, cotton balls. When caterpillar is the second stage of a life cycle of insect, when it's come and eat, we have already discussed, in all parts of the plant body, CRY protein has been accumulated. But in which form? Tell me. In protoxic form or in the form of inactivated form. So when caterpillar come and eat this bollworm, so that this CRY protein is entered into caterpillar's stomach. So in caterpillar gut I am taking. Gut means alimentary canal. If you take the gut of this insect, so this is midgut. This is midgut. Midgut have a specific cells here. These cells are epithelial cells in the stomach or in the midgut of insect. What is this? Epithelial cells. So this is epithelial cells of what? Midgut. Midgut. So what is entered into the midgut? The plant part. So look at here, this caterpillar already biting and taking the plant part. So here what is there? Plant parts here for the digestion. Look at here, and CRY protein also here. You got my point? So here which protein is there? CRY protein also here. In the form of which stage? Tell me. Inactive. Inactive, or we can also say protoxin. Also we can say protoxic stage. So when this epithelial cells of insect midgut started to secrete, what they started to secrete? Alkaline medium juice, that is digestive juice to digest this plant product. plant food material, whatever the leaf part, whatever the ball part they consume, the insect eaten, to digest this cryprotein, this epithelial cells secrete juice, that is alkaline medium. Alkaline medium juices are there. This is alkaline pH. The juices contain what? Tell me. Juice is alkaline pH. When alkaline pH is maintained, when alkaline pH is there, in presence of this alkaline pH, this inactivated protein in presence of, tell me, alkaline pH, okay, this inactivated protein turn into active toxic protein. Active toxic protein. You got my point? This is active toxic protein. Now, whenever this inactivated protein turn into activated toxic protein, it make holes on this epithelial. It is called what? Perforation. It is called perforation. That means hole formation. Small hole formation on the epithelial cells of this midgut of insect. So here hole formation or pore formation. Water entered into this epithelial tissue. Water entered into epithelial tissue. Now this epithelial tissue swell, burst, rupture. This epithelial cells swell, okay, and rupture. So when epithelial cells are swell and rupture, the elementary canal damage, insect will die. The caterpillar will die. So as soon as this caterpillar come and eat the food, biting the leaf part or ball part or what they call any part of the plant body, when they consume, immediately in the stomach that cryprotein will go, it convert into a toxin protein and it can damage the epithelial tissue of the midgut of insect will die. So that this crop is not going to be affected by any insect here. So that's how we are making insecticidal resistant plants. So insecticidal resistant plants are called BT crop, BT brinjal, BT soybean. I hope you understand. So the cry gene is giving cry protein. We are isolating this cry gene and introducing into the plasmid. With the help of plasmid, we are transfer this cry gene into the plant and we are producing insect resistant plants. This we can say BT cotton. Or we can also say BT rice also there, BT soybean also there, genetically modified organism. So with the help of rDNA technology, we are introducing bacterial gene into the plant and we are constructing insect resistant plants. And forgetting of what? Good product and also nutritionally enriched seeds and crops. I hope you understand the application of biotechnology in agriculture field. This is one of the very important concept in 12th biology. Thanks for watching.
Brilliant Qatar, your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.